Welcome to the 2025 Know Your Net's Virtual Patient Education Conference. I'm Elise Gellerman, CEO of NetRF, and we're honored that you have joined us today. This is the sixth Know Your Net's program, and we're so proud that the conference has grown every year. Today, you are among 1,000 fellow neuroendocrine cancer community members. Know Your Nets is made by and for you. By responding to our surveys, you have helped to shape this year's conference. You told us what you'd like to learn from the program, and we listened, so we thank you for that. Today, you will hear from researchers and experts on new treatments and trials and the importance of research, and you'll gain wisdom on living fully from neuroendocrine cancer thrivers. I want to take a moment to recognize the incredible community that has gathered for this event. We have participants from every state in the United States, and we're joined by people from 43 different countries around the world. 77% of you are patients, and another 15% are caregivers. That means this space is full of insight, strength, and compassion. For two-thirds, this is your very first neuroendocrine cancer education program, so we especially want to welcome you. About a third of you have been diagnosed within the past 1 to 2 years. Whether you're newly navigating this disease or further along, you belong here. This year, NetRF is celebrating a milestone. It's our 20th year of funding research and educating people with neuroendocrine cancer. For 2 decades, we've been at the forefront of advancing neuroendocrine cancer research, and we're incredibly proud of what we've achieved together. NetRF is now the largest global funder of neuroendocrine cancer research. When we began, there were 2 approved treatments for NETs. Since then, thanks to years of research, collaboration, and sustained investment, 7 new therapies have been approved. Later on, you'll hear about NetRF's research priorities and our commitment to funding breakthroughs today to develop cures tomorrow. Just as we have been driving research, we also educate and empower patients and caregivers. We are committed to helping you understand neuroendocrine cancer, make informed decisions about treatment, and know that you are not alone. In addition to Know Your Nets, we offer a number of invaluable patient resources, including the Net Knowledge Center, a patient guide, the NetWise podcast, and our video library. Visit netrf.org for more. Some quick notes about our program. Please check out the conference booklet for more on our speakers and sponsors. If you have questions for our live Q&A panel, you can submit them through Slido on the conference page. Know Your Nets is a team effort. We are grateful for the many NET and NEC experts who will be speaking today. Special thanks to our co-sponsor, University of Chicago Medicine. This program would also not be possible without all our sponsors, Exelixis, Curium, Crinetics, ITM, and RaiseBio. We start today with several presentations about breakthroughs in research and innovation. It is my pleasure now to introduce my co-chair, Dr. Xavier Koitkin. He is the director of the University of Chicago Neuroendocrine Tumor Center. Dr. Koitkin is a surgical oncologist with expertise in neuroendocrine, thyroid, parathyroid, and adrenal tumors.
Welcome all to the 2025 NetRF Know Your Nets Patient Conference. We're really excited to have you here today. It's been my 7th year as the co-chair of this event, and I've been privileged to work with NetRF for such a long period of time. I think we have achieved a lot over the last 7 years, and I believe that this year will be one of our best programs yet. We have a fantastic crew of speakers from all over the country, and we're really excited to share their presentations with you today. I hope that you will enjoy the day, and we will see you for a question and answer session at the end of this event. Thank you. And as we continue our exploration to the advancement of immune therapy, it is my pleasure to introduce Dr. Jennifer Eads, who's professor of clinical medicine at the University of Pennsylvania. She's the physician lead for gastrointestinal clinical research and a director of the UPenn Neuroendocrine Tumor Program. Please join me in welcoming Dr. Eads as she presents on expanding possibilities with recent developments.
Hi, my name is Dr. Jennifer Eads. I'm a GI medical oncologist at the University of Pennsylvania Abramson Cancer Center. And I'd like to thank the Neuroendocrine Tumor Research Foundation for asking me to present to you today on some very exciting new developments and things that are coming along down the pike for patients with neuroendocrine neoplasms. So the title of the talk today is Expanding Possibilities and Recent Developments. So the objectives of our talk today are going to first be to review the importance of the somatostatin receptor and opportunities for targeting it. It is one of the things that we've capitalized on the most over the last several years, and we'll just have some sort of brief discussion on this. But then we'll spend the majority of the talk really on immunotherapy, and we'll start by defining immunotherapy and how the immune system may be manipulated to treat cancer. And then we'll discuss the various immune-mediated approaches being evaluated in patients with neuroendocrine tumors and neuroendocrine carcinomas. So first, as I mentioned, targeting the somatostatin receptor, something that's been really critical to us over the last several years and is the foundation for peptide receptor radionuclide therapy, or PRRT. Many neuroendocrine tumors express the somatostatin receptor, and we can really capitalize on this for both diagnostic and therapeutic purposes. Many of you are probably familiar with dotatate PET imaging, which is a much more refined imaging modality that we have for visualizing neuroendocrine tumors. You can see in the picture on the left that as compared to the CT scan at the top of the picture, that the dotatate PET imaging gives us a much more refined look at the degree of tumor involvement in a given patient. And then on the right, this is a depiction of how PRRT works. So essentially what PRRT involves is that you have a radiopeptide that is injected into the blood. That radiopeptide then binds to a receptor on the cell surface, so in this case, the somatostatin receptor. It's then internalized into the cell, and we get release of the radiopeptide within the cancer cell. So PRRT allows for essentially targeted radiation therapy to be delivered to the neuroendocrine tumors. It's relatively sophisticated. So that's been one of our major developments over the last several years. And the way that we're currently trying to enhance that is largely through alpha emitters. So currently, the PRRT that most people are receiving and are familiar with involves a beta emitter, which you can see in the picture on the right here. A beta emitter kind of generates its energy that wanders a bit and is not specifically targeted towards something very close to it, but rather travels throughout the cell and is of lower impact. Whereas an alpha emitter, which you can see in the top panel, is more of a very direct focus of energy and a large hit. So we essentially get a higher linear energy transfer with alpha particles as compared to the beta particles. And what this results in is a greater degree of double-strand DNA breaks, and that allows for there to be a greater degree of cancer cell death. So this is an area that's still being actively investigated. There was a trial that was recently presented, the AlphaMedix 2 trial, that utilized a lead-based alpha emitter therapy, and they assessed for safety, tolerability, and efficacy. And we see in that trial that the response rate of neuroendocrine tumors to this type of therapy was over 55%, which is, I think, better than anything we have in any other type of treatment that we have for neuroendocrine tumors. And in comparison to NETTER-1, which was the trial that PRRT is based off of in this country, we saw a response rate of only 18%. So alpha therapy is looking promising, but this is largely an area of ongoing investigation, and there are a number of trials available throughout the United States and beyond that are ongoing at this time. So as I mentioned, we're really going to be focusing primarily during this talk on immunotherapy. So I wanted to make sure that everybody has a good understanding of what immunotherapy really is. So this is essentially an approach to treating cancer that attempts to capitalize on the patient's own immune system, where the immune system identifies a cancer as foreign and therefore attacks it. So there are multiple ways for us to manipulate this system. One is to lift any blocks on the immune system in the cancer. So some cancers, they have a resistance mechanism that allows them to sort of not allow the immune system to attack them. And this approach is largely through the use of immune checkpoint inhibitor therapy, which we'll talk about. And then the other way is that we can really try to force an enhanced engagement between the immune system and the cancer cells. And there are a number of ways to do this, all of which we will go through, but that includes antibody drug conjugates, bispecific T cell engagers, chimeric antigen receptor T cell therapy or CAR T therapy, and the use of tumor infiltrating lymphocytes. So the immune checkpoint inhibitors are the ones that are probably the most well known to everyone. There are multiple commercials on television talking about their use in the treatment of many types of cancers. And the way that these work is that there are signals that are present between the immune T cells and a tumor or other types of cells that can control if the tumor might be attacked by the immune system. And that's really depicted in these images on the right, where you can see in that panels that say without immunotherapy that there's this engagement between the T cell and another cell that sort of says, nope, you're not going to be allowed to attack. The immune checkpoint inhibitors act on these blockages and allow for these signals to be lifted so that the immune system can then attack the cancer. And you can see that as depicted on the right side of each of these images, where you have either an anti-CTLA4 antibody or an anti-PD-L1 or anti-PD-1 antibody, disrupting that connection between the T cell and the tumor cell or some other type of cell. And that release of the blockade on the immune system allows for the immune system to then go ahead and attack the cancer. Unfortunately, in neuroendocrine tumors, we don't really have much of an immune-mediated element to this disease process. So in order for immunotherapy, like I just referred to work most effectively, there oftentimes has to be a high level of mutations present in the tumor. And different tumor types can have different mutational burdens. And you can see this in the figure on the right. So cancers such as melanoma or lung cancer have a very high frequency of mutations in their tumors, whereas neuroendocrine tumors, where you can see depicted here on the figure by carcinoid, have a much lower mutational burden. And as a result, the immune checkpoint inhibitors don't really work very well on this particular type of cancer. And we've done multiple trials of immunotherapy with the immune checkpoint inhibitors in patients with well-differentiated neuroendocrine tumors, and they've largely been unsuccessful, with response rates being only 0 to up to 9%. We have had a little bit more success in the more aggressive high-grade neuroendocrine carcinomas, which behave more like some of these other more aggressive cancers. But generally speaking, we haven't really been able to make a lot of progress with the immune checkpoint inhibitors in the neuroendocrine neoplasm space. So that brings me to talking about those other 4 mechanisms that I mentioned, where we're essentially trying to force the immune system to engage with the tumor. So one way of doing this is by utilizing that somatostatin receptor that I referred to previously. So essentially bringing the immune-mediated therapy to the somatostatin receptor. So we can engineer some of these treatments to bring the immune system in closer contact with the cancer. You can see in the figure on the right here at the top that in a normal immune system and its interaction with a cancer cell, that there really is this connection between the two that does not necessarily allow for immune activation. With a medicine that can be engineered, such as a bispecific T cell engager, this is a molecule that essentially allows for there to be a connection to the T cell, and then also some kind of receptor present on the tumor cell. So in the case of neuroendocrine tumors, oftentimes this is the somatostatin receptor. And by the medication having this ability to connect to both cells at the same time, it allows for this T cell to be brought into close communication with the cancer cell, and then you get sort of engagement of the immune system. So we have simultaneous targeting of the somatostatin receptor, for example, on the neuroendocrine tumor cell, and CD3, which is a molecule present on the surface of T cells that results in an enhanced activation of the immune system in close proximity to the neuroendocrine tumor cell. So this specific example of a neuroendocrine tumor cell interacting with a T cell through a somatostatin receptor is something that is currently in development. However, there is an agent that has been approved that is looking promising in neuroendocrine carcinoma, so the higher grade, more aggressive neuroendocrine carcinomas, but is not yet sort of broken through in that area. And that is an agent called tarlatamab. So this is a bispecific T cell engager that targets DLL3, which is a molecule that's present on the surface of many high-grade neuroendocrine carcinoma tumor cells, and then also targets CD3 on the T cell. So this agent has been studied and subsequently approved for patients with small cell lung cancer, which is important for us because there are actually a lot of things that we believe are in common between small cell lung cancer and high-grade neuroendocrine carcinoma. So when we see a treatment that is promising in small cell lung cancer, we often wonder, would it have a place in high-grade neuroendocrine carcinoma? So fortunately, DLL3, which is the target of this medication, is also seen on the surface of a very high percentage of high-grade neuroendocrine carcinomas. And so now tarlatamab is being investigated in the high-grade neuroendocrine patient population. To just kind of give you a preview of what this may look like, hopefully, our hope would be that we could do as well as in small cell lung cancer. But in the trials of small cell lung cancer, we saw that there were responses in about 35% of patients. This figure here depicts kind of what the onset and duration of response looked like in that 35% of patients. These figures are reflective of two different doses that were administered in the trial. And the green triangles reflect when in the course of treatment the response was seen. So usually after 6 to 8 weeks, we see that there is a response. And then you can see from the blue lines how long each of those patients responded to the treatment in terms of the number of weeks on therapy. And some of these patients are on for over a year, and we get pretty favorable results. So again, this is only a diagram reflecting the patients who responded to the therapy. Only 35% of patients responded, but it does show what the trajectory of those patients' disease course looked like, and that looks pretty promising. When we look at the survival for patients receiving tarlatamab. So this now includes patients who both responded and who did not respond. But we see that on average, patients go without having disease progression, so no meaningful growth in their tumor, for somewhere around 4 to 5 months. And people survive for a little over a year on this medication. And you have to keep in mind that this is a medication that is given after standard therapies for small cell lung cancer had been administered. So that's not including the time that they had been on those treatments. And so compared to the other things that we have available for patients with high-grade neuroendocrine carcinomas, if we were able to achieve something similar to this in high-grade neuroendocrine carcinomas, that would be one of the most promising things that we've found in that disease entity ever. Again, with caution, this is data that resulted in an approval for tarlatamab in patients with small cell lung cancer. However, we're optimistic and hopeful that we'll see some promise of this agent in high-grade neuroendocrine carcinomas. And as I mentioned, those trials are ongoing. An alternative approach to targeting surface molecules present on a neuroendocrine tumor is through a mechanism called antibody-drug conjugation. So these drugs basically have 3 elements. There's an antibody that binds to a target on the surface of the cell. So this could be the somatostatin receptor, it could be DLL3, it could be something else. There is then a linker that is attached to the antibody that binds to it a payload that is going to ultimately be released within the target cell. And then there is the payload, which is oftentimes a chemotherapeutic agent, And when it's delivered in this mechanism, it can oftentimes be more potent than when we give chemotherapy just by the standard way through the blood system. So basically, what this overall mechanism allows for is targeted binding by the antibody of some kind of payload that is toxic to the cancer cell and localized delivery of that payload to the cancer cell. So extensive research is needed in all of these elements of the antibody drug conjugate, both the antibody, the linker, and the payload. There's a lot of very sophisticated chemistry that goes on with developing these agents, but we have currently many investigators, including several through the Neuroendocrine Tumor Research Foundation, who are looking at novel targets and payloads that may ultimately be incorporated into an antibody drug conjugate and used for the treatment of either a neuroendocrine tumor or a neuroendocrine carcinoma. Moving on then to some of the cellular therapies. So there's a couple of cellular therapies. One is CAR T therapy, which again stands for chimeric antigen receptor T cell therapy. And the way that this works is that the immune system can be modified via genetic mechanisms to modify the T cells in such a way that they will be more inclined to recognize a cancer cell. So one such target on neuroendocrine tumor cells has been a molecule called CDH17. It's a target present on the surface of a large number of neuroendocrine tumor cells. So over 90% of neuroendocrine tumors express this. And this construct has been engineered such that the T cell is modified to incorporate this construct so that the T cell will recognize CDH17 on a T cell. And this has been looked at first in mice. We did some mouse experiments where a mouse was injected with cancer cells and generated a tumor. It then received 2 doses of this CAR T therapy, and then the tissue was analyzed. So if we look over at this panel on the right, in the graph, the black line is an untreated neuroendocrine tumor. And you can see that over the days that pass, there's an increase in the tumor volume over time, like the cancer just continues to grow. However, in the tumors that were treated with 2 different versions of this CAR T therapy, both reflected by the red line and the green line, you can see that the tumor volume shrinks after that therapy. And especially with the green line in the graph, we essentially get down to a point where no tumor is detected. If you look at the slides on the right-hand side, the green dye is CDH17, which is reflective of the tumor cells. And you can see on the bottom, the green cells are completely gone. So after treatment with the CAR T therapy, it was essentially able to eradicate the neuroendocrine tumor. So now this is a mouse study, which is a lot different than in humans, but that led to the development of a clinical trial that is looking at the same CDH17 directed CAR T therapy in patients with neuroendocrine tumors. So this is a trial that is actively ongoing. It's open at 4 sites within the United States. So it has 2 elements. First, we're looking at a variety of different doses of the CAR T therapy to understand what is safe and tolerable for a patient. Once we understand the optimal dose to be administered, there is an expansion cohort where we'll then treat a larger number of patients at the ideal dose level to see if this is truly an effective therapy. In addition to this study being done in patients with neuroendocrine tumors, it is also a trial for patients with colorectal cancer and gastric cancer. But right now we're really focusing on patients with neuroendocrine tumors and colorectal cancers. So the way that this treatment works is that if a patient is eligible for the trial, they come in, they get an IV put in that removes T cells from their body. So we basically collect the patient's own T cells from their body. We then send those off to be engineered so that they now express that construct that recognizes CDH17. We then grow those in culture so that we generate a lot of T cells and we have a large dose to be able to give back to the patient. The patient then receives some chemotherapy that kind of knocks out their immune system to make space for the T cells that we're going to give back to them. And then we give back those T cells. And the hope is that those T cells, now with that engineered construct, are going to hone in on the cancer cells and allow for destruction of the tumor. So this trial is currently going on, as I mentioned, and I imagine that over the course of time, as further research identifies other targets on the cells, that there will be other types of CAR T therapies also investigated in neuroendocrine tumors. Similar to CAR T therapy is something called TIL therapy, or tumor infiltrating lymphocyte therapy. In patients with a cancer, normally the immune system will try to identify that tumor as foreign, and lymphocytes will infiltrate the tumor and start to work on trying to identify them as foreign and work to attack them. In the figure on the left, you can see the tumor cells shown in the brownish color, and then you can see that there are a variety of different immune cells that are infiltrating the tumor. And those are ones that obviously have identified the cancer as foreign and think that it should not be there. So what we can do is we can obtain a biopsy of this tumor, and we can extract the lymphocytes that are there, thinking that those are the ones that are most likely to recognize the tumor as foreign. We can then, on the outside of the patient's body, grow those lymphocytes to make more of them. We can also modify them to make them more active. And then similarly to what we do with CAR T therapy, we then give the patient some chemotherapy to knock out their immune system and then infuse these cells back into the patient in the hope that these are refined lymphocytes that are more likely to attack the patient's cancer cells. And there are trials currently underway investigating this form of therapy as well. And so the results are still to be determined. Neither of these cell therapies are available for patients with neuroendocrine tumors at this time outside of a clinical trial, but we are hopeful that they will ultimately lead to some success. So in summary, PRRT is something that we've known about for a while now, and it is evolving with extensive investigation in alpha therapy. And our hope is that we'll have even better outcomes than we already do with the current version of PRRT. And then from the immune standpoint, we know that initial attempts using immunotherapy through immune checkpoint inhibitor therapy to treat neuroendocrine tumors has been unsuccessful for the most part, but we do have hope in the sense that we are investigating bispecific T-cell engagers, antibody drug conjugates, CAR T therapy, and tumor infiltrating lymphocyte therapy. And trials are ongoing in all of these areas. And so stay tuned, and hopefully some of these will lead to a new standard of care at some point in the future. Thank you so much for your attention.